Here we are. Very confusing. Let's see.
Scott.
Hey, Scott.
Hello.
Yeah. Full screen layout. So,
Captive Desk,
yeah.
Skeptic Philosophy. Thanks for joining, everyone.
Oh, we got a few people coming. Good. So why have we gathered here? Oh, I know. Cornelius Hunter is kind of pissing me off.
He's returned.
Did you know about him before?
Not really, no.
No, I didn't either. I think this is like his first show or something.
I think I saw his channel, Darwin's God, but I think that's what it's called. But yeah, I haven't actually seen him in the flesh, so to speak, until now.
Yeah. Yeah, I looked at his 25 failed predictions of evolution, and it's like, why does he think those were predictions? I don't know what he's talking about. It's just crazy shit. And I got off on the protein
of such predictions.
Yeah, I'd like to see where evolution predicted this and it failed, and oh my god, over and over again. It's like I don't remember these predictions, so I don't know what he's talking about. Kind of crazy.
I will be fair to him and say that when I read it, there were a few points that I wasn't familiar with from creationists already, and actually some interesting stuff I learned after kind of digging into what he was actually talking about. So I will get kudos to him for that compared to other creationist writings. So,
he's a smart guy.
At least he's original.
He's smart and he's educated, but he is just stupidly biased. So he's intelligent but stupidly biased. And his whole deal is that evolution is a religion, and he's got this whole historical idea about how our thinking got off track because we were thinking about God and we were trying to figure out what God would do. And in that process, we came up with naturalism, because some people decided that God didn't want to get down and dirty in the dirt. And it just goes on and on. And it's like none of it has anything to do with 2025 biology and evolution. And he thinks that it's clouding our thinking from the 1800s, right?
Yeah.
Which is his whole deal. But he
reviews that biologists today are reading things from the 1800s, to be honest.
I know. He thinks we're the bias and the way our culture evolves. So he's one of these guys that believes that science is based on culture. That our evolution is based on culture.
Which I mean, to be fair, it partly is. So.
Yeah, but there's
he's claiming.
Yeah. Naturalism is not based on culture. It's based on what you do every morning when you get up out of bed. Like when you get up out of bed, you do not pray to God that the floor is going to be there. You step on the floor and you expect gravity to basically hold you down.
Right. We follow methodological naturalism because it works, and it's worked for so long. So.
Yeah. Well, it works in your everyday life, basically.
Right. Yeah.
But anyway, so he's going after this Kunin paper in this latest video, and I have so damn many bookmarks that it is disgusting. It's absolutely disgusting.
Like a chromosome there.
Where's that?
on the all the bookmarks you got there, all the little.
Oh,
Yeah.
yeah, it looks like a chromosome. Anyway, in silico evolution of globular protein folds from random sequences by Wolf, Kunin, Babajanyan, and I don't know how to pronounce that.
Oh, it's fairly recent.
Yeah, it's real recent, and it's kind of a neat paper. And what they do is they start out with, I think, 100 amino acid sequences. I can't remember. Short sequences, and they keep mutating them. And then they run them through AlphaFold, and I think Rosetta Fold too. And they basically determine how they fold. And then what they do is their selection criteria is if it forms a globular protein, meaning that it's more likely to not hurt the cell if it folds up, because basically the water soluble parts are on the outside, and I forget exactly how. Yeah, they're not likely to stick together and form these globs. So basically, he uses a real weak selection criteria, and he comes up with, and what they found out is that sometimes within 0.2 mutations per site, I think, that they can come up with valid folded proteins, and they've got a whole bunch of them here. That they just a whole variety down here. Wow. Yeah, just a ton of them. And what they found out is that the possible functional density in the protein space is higher than we thought. That's what the whole paper was about, showing what the density is, how close these folds are to each other. Things that will validly fold.
Right.
And then let's see, and I haven't finished the paper yet, but they found like some of the things resembled proteins. Some of the motifs resembled functional proteins that are in our genomes today. I think they found like 1 or 2 in one run or something, and they were surprised to find that, because protein space is really big. Anyway, so that is Kunin's paper. And let's see here. Go back to this, go back to that, and go back to full screen layout. Okay. And he wrote a letter to the journal, and they politely blew him off.
Hunter wrote a letter to the editor?
Yeah. He wanted it published basically as a critique of this paper, and they blew him off because they said that he really wasn't contributing anything that they were interested in. And Hunter doesn't understand. He thinks they blew him off because he was criticizing evolution.
Why they always think that?
Yeah, they always think that. Yep. Anyway, but this is kind of why they blew him off, because they probably know who he is.
Hey, Mike, you might want to introduce Andrew for future generations. You might want to introduce Andrew.
Oh,
Skeptic philosophy wasn't familiar with him. And,
rational empiricist. I don't know if you want to put your last name out there, but
yeah.
Anyway, Andrew.
Pretty much everyone else knows who I am at this point. So.
Yep. You're a student of evolutionary biology, right? Have you decided on a major yet?
Yep, ecology and evolutionary biology.
Yep.
Bingo.
Okay. Bingo.
Currently working toward my bachelor's in biology.
Yeah. Yep. Have you started messing around with NCBI and UCSC yet?
Oh yeah.
Gene? Okay. You could probably fill in some of my blank spaces, but I have learned a lot about how to operate it, how to get from one thing to another. I
still think you're probably more proficient in some of that than I am, but
yeah, we could get together sometime and compare notes on it. Maybe I'm sure we
can
learn
from each other. For those things.
What's that?
I wonder if there's a subreddit for for users of those
I bet there is.
apps.
Yeah,
or
I bet there
programs,
is.
whatever you wanna call
Yeah. Okay, so I'm going to play a little bit of this. This is the bogus part.
Is a very subtle problem, an example of a very subtle problem in the evolutionary literature, which is actually an enormous problem. It's bogus science, and it needs to be understood, but it's not simple to understand. So that's what I want to get into, but I want to give some background first.
Okay, bogus science. That's what he's calling Kunin's work. And
Is
he
that
does
a
it over. What's that?
Is that a technical term? Bogus
I think so,
science?
yeah.
Do all
Yeah,
the
bogus science.
philosophers of science refer to it like that?
Yep. Yeah, I wonder, I don't know what's in between all these things, but let's see if he says anything valid here. I don't think he does
With a
here.
drink of water, I will go there.
Oh.
So there's a key concept, which is not only important for understanding this paper, but in general, anybody who wants to understand origins and evolution and the impact on our culture and everything that's happening needs to understand this one concept called teleology. It's a word that a lot of people have heard.
Yeah, okay. Blah, blah, blah. He's going on about teleology. Yeah. And Rebecca was talking about Dennis Noble, and I think he talks about Dennis Noble. And Dennis Noble does not think that there's teleology in evolution. Dennis Noble believes it's teleonomy, which is kind of the appearance of teleology, basically. And they misunderstand Noble. Of course, Noble, his rhetoric, he plays to the public, sort of.
Yeah.
He's an old stinker, is what he is. He's just, I think he's having fun with just pissing everybody off all along the line.
It's definitely not a mainstream view.
But he's got some valid points for extended evolution, which I agree with, and that is the basically looking at it in different hierarchies and looking at causation from top down and bottom up and everything in between. And that's kind of important to understand. But most of his work is in the immune system, and there's a whole lot of teleonomy there, but it doesn't really apply to everything else, and Noble sort of oversteps. Yeah. And anyway, it's interesting.
It reminds me of that one quote creationists like to cite, where it's either Francis Crick or Richard Dawkins saying it appears designed, but we have to remember that it was designed by natural selection, basically. I forget what the exact quote was, but it kind of reminds me of that.
Yep. Yeah, and we design things, and we are natural, right? We are naturalism in action, and we design things. So when we design things, it's still naturalism. There is no teleology.
Yeah.
It's kind of interesting. Anyway, skipping ahead here quite a bit. I got a little bit about Kant's.
Millions of species to arise. All those changes had to have happened by themselves, by chance, according to evolutionary theory. So we need to understand what the theory is saying here. So it's not this tornado forming the dock is not just some academic example. They're insisting on this, and you can go back to the Epicureans for this.
We're not going to do that. Chance, basically. Okay. No, it wasn't just chance. Dice were not rolled.
Really,
there was
fascination with ancient Greeks' ideas about evolution.
He does, yeah, he does. Basically, it was causation is not well described as random or chance. Okay, random is a word that we use when we have an independent variable, basically, like we roll dice and we don't know everything about the physics of the dice, the hands, the error in the room, all of that stuff. And so we claim it's random, right? And because we can hit it from a number of different systems, right, or a number of different circumstances, it will come out with a probabilistic curve because we're basically going across a number of different pressures, throws, whatever. And we use the word random basically in statistics, and using it in evolution is just a little bit off. Okay.
Right.
I mean, if
Yeah,
we're going to simulate
yeah.
mutation, right? Anyway.
Yeah, usually the most you can get is a probabilistic distribution in contrast to a deterministic one. Like true randomness is very hard to find in nature.
Yeah. Yep. Yeah. I think pi, they think it's random, but the digits in pi, they seem to be random, but yeah, we have a hell of a time finding really good random data.
Yeah, it's the other thing. Like maybe we're really good at finding random patterns.
Yeah. Yep. Oh, and going ahead here, and I guess we'll play him for a little while, see if we can get
where this originally came from. An example they used was teeth. What are your teeth for? And you say, well, that's obvious. The teeth are for chewing. Well, they would say, no, the teeth are not for chewing. The teeth are good at chewing, and so they persisted. Sounds kind of a strange thing to say. Why are they saying this? What's the point? Why are they taking this kind of weird position? This is kind of is this a weird ancient Greek thing? No, actually, because this persists to today with evolutionists. The reason they're saying that is because if you say that the teeth are for chewing, you're implying intent, and you're implying a designer and a creator and all those things. And therefore, you are assigning to that creator toothaches and a whole list of other things that we don't like about the world. So there's a whole raft of arguments of why God would not have created this world. God would not have, should not have, could not have, didn't. We know that God didn't create this world. And some of them deal with the evils and the disutilities and the pain and the bad things, the diseases and so forth of the world. Some of them deal with doctrines of God. Some of them deal with doctrines of man. So it's all over the map, but there's more than a dozen of these arguments that are religious theological arguments about why God would not have created this world. This forces the Epicureans into this.
We'll go back to that again.
I mean, what I really find weird about this argument is, like, of course, he doesn't say anything positive about, or doesn't make a positive claim about what the designer would do, because I mean, you see other creationists, though, they do make positive claims about, like, oh, they would design it this or that way, especially when you're comparing it to human designs. Like, that's what Donnie likes to do a lot. So it's like, okay, cars, the car analogy. But like, if you're, obviously, when you compare life to human design, it looks nothing alike. So it's like, based on that one criteria, we can say, or based on known design, which is human design, life doesn't look designed, at least when you look at it in a lot of detail, not just superficially. I just, his whole argument here is pretty backwards.
Yep. He okay, saying that the teeth are for chewing, right, is an artifact of our language and what we do, and it actually expresses what we needed to express, and that the function basically, teeth chew things, right? But there's no reason why some other system couldn't have come up, like maybe little acid sprayers or something that blasted the food apart or whatever,
or something like that.
Yeah, but teeth are like the most likely solution to come up, because of bones and all of that stuff. So So he's complaining that we use the word for in our papers, and he does that quite a bit. There's somewhere he talks about the elite. Let me see if I can get to that.
Position of saying, no, you cannot say that the teeth are for chewing. They must, like that dock, that unrealistic dock example where the tornado forms a dock. God, these teeth cannot be intended for. They must have arisen by chance. They used their mechanism was randomly swerving atoms. Okay, so randomly swerving atoms created everything in the world.
Hey, not much different.
Randomly swerving atoms, yeah. Oh my God.
Different than what's being proposed today.
Exactly, not much different. Random. Now we have random mutations. They had random motion. We have random mutations.
I don't think Rebecca has much awareness of what's actually being proposed today.
No, I don't think she does. She kind of admits it later. She starts talking about proteins, and she doesn't seem to really get them. And she says she doesn't like the word.
I really hate to say this, but like I feel like she's become more smug, confident kind of attitude lately, especially when she's talking about or people in the comments like, oh, you don't know the literature, you need to study more science. It's like she's getting at that really point that I wish she'd kind of back away from.
Yeah, she's become more stubborn and kind of smug. Yeah. I don't know. I think that's from hanging around YouTube. It's starting to get to her, and she needs to go and talk to her pastor and have a good sit down with him and kind of.
I mean, I know she's hung out around Donnie's channel, so maybe partly that too.
Yeah, could be that. Yep. I wonder if I can get the transcript up here so I can see what the hell I'm looking at. Let me see here. Oh,
yeah, his whole argument here, it's just a shorthand. Like, obviously, chief evolved for chewing is a lot easier and more concise to say than teeth are very good at chewing and gave the organisms in the past, the ancestral organisms in the past, an survival advantage, so they had more offspring and passed that teeth trait on to their offspring and so on. Like, it's just much easier and it gets the point across. So like he's kind of arguing semantics here.
But it isn't always about more offspring. Sometimes it's about basically something that just happened in the genome, basically, or in the population. And then later on, it became functional and it became useful.
Right,
and therefore it did a sweep.
Perspective here.
Yeah, neutral. Like there's a lot of neutral things going on.
Right.
And choices, that sort of thing. Oh, let me see if I can find. I want to find the transcript.
And obviously, that's a lot different than what he's talking about with this tornado in the junkyard. Like nobody's claiming there was a creature with no legs at one point, to use an example, and then a few generations later there were randomly creatures with legs. Like, no, that's not what we're talking about here.
Yep. Oh, let's see. Not for teeth purpose. Why are my bookmarks off? People hear this. So yeah, I think I'm gonna just play a little bit. It kind of shows
fast forward to modern times and Charles Darwin, and even before Darwin, but
modern
Darwin and after Darwin, this was elucidated equally with the evolutionists. They made it very clear. It's not for, teeth are not for chewing. Everything arose, as Darwin explained, by random change that was then inherited and retained by natural selection. So it is core to evolution.
Yeah, natural selection. They don't seem to understand how that plays with the neutral evolution for selecting these traits and the traits that survive.
Well, it's like they always isolate one part of it, like it's mutation, selection, drift. They don't think about it holistically.
Yeah. Yep.
Evolutionary theory, anti-teleology. It is an anti-teleological worldview, not a scientific finding, theological view, theological mandate. Anti-teleology, nothing can have a purpose or reason in this world. And so I hope that kind of gets across this idea of
this is, our thinking is so human. You know, we think we are a kind of animal. We have a premotor cortex, and we have this frontal lobe, which is an exapted premotor cortex, and we can plan things in the future. So we can say, oh, I'm going to sharpen this stick so I can poke your eye out, right?
Yeah.
So the sharp stick is for poking your eye out, right? But we are simply a natural process working to do that, albeit much more complicated when you have to, now you would have to track everything causally in the brain to figure out exactly how the stick got sharpened and all of that. So we're up in the hierarchy, right? That's where the extended evolution is coming in. Now that has something to do with it all, right? And so it's causal downward, that sharpening that stick and poking the other guy's eye out may well shape the course of evolution, from a top hierarchical, a higher level in the hierarchy, right? Yeah. Having causation downward.
Niche construction.
Yeah, that sort of thing, right? So this whole thing about teleology, teleology only exists when you have a mammal like us with a premotor cortex and a frontal lobe where you can decide on, you can sit around and plan a movement, that sort of thing. Like a cat will crouch backwards so that he can jump, right? And catch the mouse. And he's sitting there planning to catch the mouse.
Tools, so.
What's that?
I said certain octopus species use tools too, so maybe talk about them in a teleological sense.
Yeah. And these guys, they have this teleology thing. It's like, because they believe in this God thing, and they're very confused about that. Hunter is very confused. Like he said, God isn't going to get down and make the finches beaks. He's not going to get down in the dirt. Later on, he says, of course that wouldn't happen. God created something like epigenetics, right? Is what he claims the beaks are all about in the Galapagos. And he's saying that, God created the epigenetics. So God did that part. That was God's intention. That's God sharpening the stick. But then the actual beak transformation was because of epigenetics, not, in other words, it's the same as old Earth. What is it? Old Earth. What do they call it? Old Earth theology or Old Earth evolution, that sort of thing.
Like progressive or theistic evolution, progressive.
Theistic evolution. That's what I'm looking for, right? Except that these guys, they want to move the timeline up about what God did. God created man separately from chimps, and he created all these kinds and all that stuff. But he's still saying that God created certain things like epigenetics that ended up playing into evolution naturally. Okay, so it's weird, but they're choosing a point where they think God lifted his fingers up and quit interfering and let the thing roll naturally. And their point is like 6,000 years ago, which is ridiculous. You see what I mean?
Yeah.
So their thinking is so stinky with the Bible. It's just horrible. It's just yuck. I don't know. I'm going to go ahead here. Let's see. There's some more interesting stuff later on here, I think. Let me see here. Where are we at? I got a bookmark probability. Let's see here. There we go.
Hundreds, sometimes even thousands of amino acids lined up in a chain.
Oh, here we go.
These are created in the cell using the genetic code, and the information coming into the code that's going to create this sequence of amino acids is from the DNA. So it's a gene in the DNA coming in and coding for this string of amino acids. Now, there's 20 different amino acids that are used typically in the cell to make proteins. So you think of it as basically like a line of letters where each of the 20 amino acids can be represented with a letter. So you have 20, an English letter. So you have this kind of string of letters, and that's that string of letters that determines how this thing is going to fold up and what kind of a protein it's going to make.
Yep.
That sequence of amino acids is critical and crucial. And that's determined by the gene that's in the DNA. It determines the sequence of amino acids. So evolution has this problem of how could this evolve? If you think of it, okay, there's 20, I said there's 20 different amino acids. And let's look at a protein of 100, a relatively small protein, 100 amino acids. How many different possible sequences are there? Well, at the first one, there's 20 different amino acids. At the second one, there's still 20. At the third one, there's still 20. So it's 20 times 20 times 20 times 20, all the way to 100. That's the number of possible sequences that you could have. 20 raised to the power of 100. That is an astronomically large number.
10 to the 130 or something like that. Anyway, so we're going to talk about Douglas acts, right? So he acts like there's a target protein because he can't stop thinking teleonomically, right? So he thinks there's a target protein and evolution has to get there. Evolution has to get to this. Well, the Kunin paper shows us that very small peptides and foldimers, I think they call them foldimers, maybe that's RNA, very small peptides do have a function. They're chemicals. They do something. If you take two amino acids and join them together, that's still a molecule and it's still a chemical. And if you take like a aspartic acid and glycine, okay, you're going to have a chemical with a carboxyl group on one side, and it actually that would be two carboxyl groups, I think. Anyway, it would be acidic, right? And it would do something. It would have a chemical reaction. Now, if you take a string of about 20 to 100 protein amino acids and hook them together, they tend to fold. And that's what Kunin was showing, is that fold space is a lot denser than we would have predicted. Or, well, we had nothing to predict it with, right? We're exploring this space and finding out what all these small proteins did.
Bigger than we would intuitively expect.
Yeah, there's like more folds and more possibilities of active proteins.
See, that's the thing with science is intuition gets thrown out the window because so much reality defies our intuitions.
Why? Okay, space-time. Yeah.
Intuitively, you would expect objects with different masses to fall at different rates, but hey, that's not the case.
Yeah. Well, I think later on, the skeptic philosophy, later on, Hunter says that God didn't do that part, that he just did the epigenetics part. And the other thing, they think epigenetics is something that's separate from DNA.
Right.
The DNA is why there is epigenetics, basically. So
they kind of just use it as this magic wand to
put
in just enough evolution, or well, they won't call it evolution, but just enough evolution to make it work, but not too much so it's too obvious.
Yeah, in order for a sequence of DNA to get methylated or whatever, in order for epigenetics to work, there have to be proteins, and those proteins came from the DNA, and they were regulated, and the amount of protein that's there is going to determine how much methylation is there, and how that thing I did this morning, Scott, where I showed you how the chromosome territories were created.
Yeah,
that's all based on the DNA, and to some extent is based on the maternal cell, right? So there's a little bit that's passed on from the maternal cell, but mostly it's DNA that's going to determine epigenetics. Epigenetics is just a complication in the nucleus, is what it amounts to.
An extra layer of complexity, but it's not separate from it.
Right. You modify the DNA and you're going to modify the epigenetics of the organism. So it's not some magical other thing that basically is like evolving all by itself or some spirit animal. I don't know what the hell they think. Anyway, Rebecca's going to say something here about proteins in a little while, which is kind of cute.
Number, you can't really even get your head around it. It's so big of a number. Evolution, the evolutionary process somehow has to search around and find the right sequence of amino acids that's going to code for this gene. It's going to fold up in the right position.
It does not have to search around.
It's not a drunkard random walk kind of process.
Yeah. It's not looking for a particular sequence, basically. Okay, the cell is either going to survive or it's not going to survive, right? And if there's a mutation and the cell doesn't survive, then the cell doesn't survive, or else it does survive. And if it does survive, then that's going to stick around. It's just, they're not looking for a particular protein. It's like, we got to look for this protein that's 1,200 amino acids long. It's like, no. You see, their thinking is stinking with God. Basically, it's filthy with God. They can't get around it.
And do the function that it's supposed to do. You know, there's this Monty Python show, this Monty Python skit of hide and seek, where they're playing hide and seek. Some people may have seen this. It's a global hide and seek.
Oh shit!
So they start out in England, and it's okay. I'm gonna count to 100. You know.
Okay, yeah, you can hide anywhere in the world, and they got to go find them and stuff like that. And he talks about Monty Python about 16 different times, and I just want to bypass that part.
That is a hilarious skit. That is what it's like for evolution to find a protein sequence.
No, it's not.
There's very few answers in an enormous search space.
Oh, there are more than a few.
Yeah, there's a lot. I mean, it's and the Kuhnen paper talks about this a little bit, that basically they found totally different structures that look like they'd be motifs that do some chemical reaction, but they're unlike anything that we have today in any life form, in any life.
Oh, nice.
So there are a lot of other ways that it could have happened. So
right, we kind of stumbled upon this particular one.
Right. So the Monty Python thing is the guy, you're playing hide and seek, and he I can't remember that skit, but anyway, you can go anywhere in the world and hide, right? And then you got to set out to search for him, right? Well, that is not like evolution. Evolution would be like, okay, so he goes out and hides somewhere, but the first thing you do is you find a footprint pointing in a certain direction, and that footprint sends you on that direction, right? And the next thing, you see a branch broken, so you're following a trail, basically, right. And that trail is natural selection. So natural selection lays down a trail.
It's a Markov chain, basically.
And along the way, you're making predictions about what to expect upcoming based on what you've discovered and learned previously in the path that you've taken. It's not a blind path. It's you can construct some facts together, build models, and predict. Sure, surprising things are going to be found out, but you can still make predictions and move forward and be more likely to discover the next footprint.
Yep. Yeah, the neat thing is that, okay, if you are a human and you're looking for somebody and you're going to follow their tracks, or basically all kinds of clues to find them, what you're doing is you're taking each piece of information and you're putting it into your memory, right? And from that memory, you go on to the next step, right? And that's how a brain would work, and that's how a mammal would do it. But with evolution, what happens is that each step, there is an extant population of living organisms that are functioning. Okay. And then it can only take the next step along the path. So every population that existed in the past is kind of like a bit of memory of that, of that, the whole sequence, right? Where it goes. And that's a better analogy. Yeah.
Yeah, the immediate or future changes depend on the immediately ancestral conditions, basically.
Yeah, and that's the right way to think about it. The way these guys are thinking about it is just totally foreign, and they don't get it.
And
I mean, they can't.
those minute changes have to be functional with selection.
Yeah. Okay, I think Rebecca's part's coming up here.
20 to raise to the power of 100 is the number of possible sequences, and how many of them are going to actually create a protein. There's thousands and thousands, millions and millions, but it's a small number compared to the space that they're searching.
Yeah, and can you tell people more about like what proteins are, how vital they are? Like you can't have life without many proteins working together. So can you describe proteins a little bit? Because I actually hate the word protein. It doesn't capture.
I thought that was so cute.
Okay, what term would you prefer then?
Yeah, I know. She just hates the word protein.
Exactly what we're talking about. So can you describe?
I think she'd like God's hammer better.
Or
maybe God's
like
hammer.
Nano machines or something like that. Probably.
Yeah.
What is a protein?
Yeah, and you want to probably go to the internet and search on protein, just look at examples and stuff to see how cool they are. But yeah, proteins perform a lot of different jobs in the body. Without them, you die instantly. When you're digesting food, when you're all kinds of jobs. When you're synthesizing chemicals, when you're making proteins, you have to have proteins that help do that job. So they are basically agents. They're little machines going around doing jobs. When you need to access the DNA to copy a gene, it's a protein machine that slides along the DNA, opens up the double helix, and makes a copy of one of the helices. So that's a protein machine. When you edit that copy, that's a protein machine with many, many proteins. When you digest food, when you're eating a piece of bread, you're bringing in this bread, and that's going to be converted to energy, chemical energy or potential energy that could be used to think, to walk, to move, to do all the things we do. It's proteins that are doing the.
Yeah, he kind of goes on for a while here, and I'm not sure where he goes next. But the stuff that Demeter and I did basically concerned our small RNAs and small peptides. And the interesting is, so it's the RNA peptide world sort of thing. Right. So, they're going to go on and they're going to start talking about abiogenesis, and they're going to conflate it with evolution, which is really frustrating because you can't pin them down. They're like, okay, how did the ribosome develop and all of that bullshit, right? But the RNA peptide world hypothesis is probably, it's kind of my favorite backbone for this whole thing. And the peptides stick onto certain RNAs and they stabilize them. And those RNAs at some point can create that peptide that stabilizes them, or peptides like that stabilize them. And you're looking for kind of like a needle in a haystack, but it was a hell of a big haystack back then around those vents, and maybe on shores or wherever it developed. And, we don't know how all that happened yet, but we've got some damn good clues about how it happens. For one thing, I think they found out that in the RNA, the last 4 nucleotides determined which amino acid was going to stick onto it. So they found the genetic code actually in the chemical structure of RNA. And that was Shostak, I think, or Shostak.
okay.
Yeah, Shostak. Yeah, he found all this really good information that basically the end of the RNA, this arm that sticks out, actually had the genetic code in it. But it wasn't on the part that reads the RNA, right? It was in a different part. So it was a different system that created those first proteins. And those first proteins were very small, but they were chemically active. And all of that is like either pre-cellular or right around the time that some sort of cell formed. And that cell could have very well
ribozymes.
Yeah. It's really interesting when you like. I ran a bunch of simulations, and every time you ran a simulation, you'd end up with different sets of proteins and RNA and stuff. And of course, we didn't have the tools that Kunin had in this thing that he did, which would have been pretty cool. But yeah, anyway. So let's see.
Proof of concept of what other biologists are doing.
Yeah. Yep. She hates proteins. Okay, linear flailing. Okay, I don't know what the hell that one means. I'm not sure. I'll go ahead and play that part.
Chain of amino acids, as I said.
Oh, here we go.
As that's created, it's created one at a time. You know, one amino acid to the next.
Oh, this is good.
To the next, to the next. You're tacking on these amino acids. There's hundreds or thousands of these things, and it's flailing around here in the cell as it's being created, and then it folds up. It may be folding up as it's being created, but in the end, it's folding up into some sort of a glob. At least the globular proteins that do these jobs. You have this glob. When it folds up, you're going to have a lot of amino acids that are neighbors that were not neighbors to begin with. So there's an amino acid here and an amino acid here, and now they're neighbors. And we call those long-range contacts. So long-range because they are long range away in the primary sequence, but in the folded up structure, which is called the tertiary structure, they're neighbors and they're actually interacting. There's a chemical interaction. So we call that a long range contact. And the theory of, so you have these long range contacts. And so when you have a globular protein, there's going to be a lot of long range contacts, right? There's going to be, a lot of these things because they're now next to each other, a lot of neighbors. So it is.
So he knows that much, right? So he got that much right. These long range contacts. But what he says next, and I'm hoping I have the right part. What he says next is kind of amazing.
It is a given. It is a no-brainer property of globular proteins that they have a lot of long-range contacts. That is a given. Whereas a protein that is formed from a random string of amino acids isn't going to have very many long-range contacts. They may be flailing around, maybe unstable.
Okay, that's false, and that's what the Kunin paper is showing. Just because it's a random sequence doesn't mean it's not going to have a lot of long-range contacts, right? You could create, and that's what Kunin did. He created all these short sequences, and then they started mutating them, and they found out that in not too many mutations, they ended up with folded proteins that had a lot of contacts. So what they're showing is that in that protein space, there are more globular proteins than we suspected, basically. And Hunter really hates that. He doesn't like the results of the paper.
It may form transient contacts and they go away, and so you can have some long range contacts, but you're not going to have so many.
Why? I'd like him to tell me why. Because because it doesn't fit his critique or whatever of the paper.
Yeah.
Right. Yeah. Yep.
As you have with a globular protein. So you need long-range contacts. So if you're going to evolve a protein,
here we go with the teleology again. If you're going to evolve a protein,
and these genes randomly evolve until they finally form a protein, as those mutations occur one at a time, they're going to tend to result in a resulting protein that has more long-range contacts than before. If evolution...
That doesn't make any sense, because it could mutate to not have that contact, or it could mutate to have that contact. Now, depending on which amino acid changes, okay, it's going to change the conformation of that protein. And there's no directedness to this, right? We're poking around in that space, and we could end up, with something that has less contacts or more contacts. There's no way to, there's no direction for that. And he seems to think
predict that ahead of time. Yeah.
Yeah.
Evolution is true. That's somehow going to happen. The problem is, as you add more long-range contacts, you're still at the half a wing problem. So now you're like this. It's still not going to do anything for the organism, unless you just get wildly lucky. Even if you formed a full protein, by the way, it might not help the organism.
The classic, what good is half a wing that he's talking about?
Yeah. And that's the thing. Now there's like I've been poking around in the genome quite a bit lately, and there are a lot of pseudogenes, and there are a lot of really small translations that happen. And there's just all these parts and pieces that probably do not contribute in any great way to function. But every now and then they lock onto something and they cause something to happen, right? And like there's one really good example with that pseudogene DDX11L2. Apparently, if it is produced in greater quantity, basically, if it's upregulated, it can have an effect on sepsis, whether or not you survive having sepsis. It's one of like 4 genes that they found that seem to have an effect on it. So, I mean, all of these things are going to attach to something and do something chemically that overall is going to affect the life of the cell and hence the life of the organism. And if you have all of these different pathways that are half formed and some dead ends or hundreds of dead ends and whatever, every now and then one of these pathways is going to connect up. And it's going to connect up at a time when the environment changes for that species so that that particular gene becomes the only one surviving. And that's how evolution works.
How upregulated did you say that particular gene was before it became functional?
I can't remember if they may have found out exactly, but I'm not sure. But they didn't really talk about that one in any detail. They just suggested that it was when it was upregulated, when they found more of it in the organism or in the human, that these people statistically seem to survive sepsis better. So, but I mean, there's a lot of stuff like that.
Yeah, just because I know if a creationist listens to what you just said there, they're going to take it out of context and say, see, there's no junk DNA.
Oh, yeah. Yeah, they're going to love that one. That paper just came out in 2025. I just found the very first evidence of any functionality at all of DDX11L2. And of course, it isn't something that any of us need unless we get sepsis. And then it might be a good idea to upregulate it. But who knows? There were 3 other genes that they found that they studied. And of course, it's a new paper. And we don't know if the research is going to stand or not. We'll see.
Right.
But
yeah.
I like it because that's how evolution happens. Yeah. Now, if it wasn't sepsis, if it was something else that basically would affect our reproductive success, then it would start to spread more seriously in the population, right? Or if something happened where if everybody in the world got sepsis for some reason, right? Then only the people that upregulated that particular gene would survive. That would basically be a genetic sweep, and that gene would end up in the entire population. And it's just a really neat example of one chemical glomming onto another chemical and having some overall effect. I'd love to know more about it, but anyway, it is proof of evolution. It is not proof against evolution. This is how evolution works.
So it depends on what the protein does. It's a very difficult problem. How are you going to benefit the organism? It needs to be a protein that it needs. So
I just explained
you're
that.
evolving this gene, you're introducing mutations. If you're really lucky, the resulting amino acid chain starts to look a little bit more like a protein, starts to fold up a little.
They're all proteins. This is where I started to doubt him. You know, I mean, they're all proteins.
Yeah.
Did you get that?
Yeah, it's not like like some of them may not be functional folds of proteins, but like, yeah, they're still going to fold up eventually.
And they're still proteins. They're still going to do something. The thing is, if you have a protein that's doesn't fold well, basically doesn't glob up, it can end up screwing up the works, and so that would be selected against very quickly. And that's why Kunin used the criteria that he used, basically, because
globular protein specifically.
Yeah. They tested them and they rated them based on how many contacts they had, that sort of thing. And so he had a real, and Hunter, that just went right over his head. He said, "Oh, that's nothing like the environment or nothing like evolution." And it's like, no, Kunin wasn't trying to simulate evolution. He was trying to show something about the protein space. He missed the point. Yeah, he keeps thinking that Kunin was proving evolution or something.
Well, it's like they always think this like that. Oh, this new paper is trying to prove the whole theory of evolution when it's incremental discoveries building atop one another.
Well, yeah, it's embedded in their thinking that they think that they can produce one quote from some asshole back in 1980 at a conference, and it disproves evolution. Or they could pull up one paper or one little place where somebody will say, well, this is a problem for evolution, like that chromosome 2 thing. All the things that they came up with were these like little one-off whiny complaints. Oh, the DDX11, the anchor D gene. And they think it just destroys the entire theory for some reason. They think one little tiny piece of information is going to prove God. It's ridiculous. They're not looking at the big picture at all. Anyway.
A little bit.
Yeah.
And maybe there's more long-range contacts, but there's no fitness advantage. You're not getting a better.
Why not?
More progeny.
Yeah, why not? There's no fitness advantage. Okay, if there's no fitness advantage, then it's neutral evolution. It's not going to go away. Okay, that protein or that gene is not going to go away just because there's no fitness advantage. It's going to hang around in the population for a while.
It may drift out eventually.
Do you think he's just?
It may drift out. What Scott?
When he says fitness advantage, do you think he means purpose?
Yeah.
Is he thinking, well, it has to have a teleological purpose in order to have a fitness advantage?
Yeah, they're looking at everything through the eyes of God, designing things on purpose. Teleonomy, basically. We got a nice thunderstorm going on here. Pretty cool.
From that organism, it's got this half a protein floating around.
Half a protein.
So
tenth of a protein.
Again, this problem, you have to have something that's close to actually a real protein to benefit. What are they doing?
What is that?
Yeah, I know. What the fuck? He's a biophysicist. I don't think he really knows a lot about this stuff. I mean, he knows quite a bit, but I don't think he's a molecular biologist, or he has an approach to that.
You, sir, are not a true globulin, you see.
Yeah.
The right folds.
Yeah, you don't have the right folds. You're half a globulin. Anyway.
What do they do in this paper? This Eugene Kunin paper.
Here we go.
Can I, before you go into what did they do in this paper, can I just ask, because I can just hear some people object and say, well, maybe it wouldn't help if you had, it's not quite a protein, but you've got so many tries. And so what if you have all of these like partial built proteins and you've just got so many tries with so much time and evolution that, hey,
No, that's not the
so
argument.
is that a problem?
Is that a problem? So I think it's 10 to the 40. There are 10 to the 40 single-celled organisms in the ocean.
Oh, wow.
And
Yep.
we're looking at 3 billion years, basically, or 3.4 billion, it's 3.2 billion years of 10 to the 40 organisms that are reproducing and dividing sometimes more than once a day. So, I mean, that's quite a bit of tries. So,
Right. Or like what you were talking about
Like,
earlier too, that you don't need to
you know,
sample
it-
the whole morphospace, protein
Right.
morphospace to get a
Yeah.
a functional result.
yeah, I think going back like to the first billion years, I think there were a tremendous amount of proteins and protein families, and they kept getting called out, basically. So there were a lot more, and they were a lot smaller back then, in the old days, the good old days. And basically, they kind of funneled down, and certain species survived and became the star predators or whatever.
Yeah, it might
But I
have been
think
the
that,
same thing with like pre-Luca organisms or
yeah,
competitors of Luca.
yeah, yep. I mean, there were probably there's just an astronomical amount when you run the numbers with that many single-celled organisms, dividing that rapidly. There's a lot of tries, Rebecca.
is it damaging to have half proteins floating around in a cell?
Because they're
Yeah,
not
so
literally
you don't really have nearly as many tries as you need. So
Oh,
this is
right.
where this astronomical number needs to be appreciated, and it's hard to appreciate this. We can't get our mind around it. But the number of the search space, again, it's a global search that the Monty Pythons get.
No, it isn't.
Yeah.
Yeah, he's claiming that he knows all of the math involved in that 3.2 billion years, right? That he knows all of the ecosystems, all of the little separate environments. He knows all of the life forms. He knows all of the chemicals. He knows everything. He's got, and he comes out and says, "Oh, they don't have enough tries." Like, we don't know that. None of us know that, right? If we were to model this, it would take a computer the size of the goddamn solar system, and it'd have to be a quantum computer in order to model this, right? So, I mean,
He's just assuming again that most of these folds are the vast majority of them are non-functional in any capacity.
yeah, that's the other thing. Yeah. He's assuming that the protein space is not dense with function. And
Who's
I
he?
think
Are you talking about Coonan or are you talking about Hunter?
Hunter.
Okay.
Coonan showed that it was a little thicker than we thought with his simulation. And
Yes,
that's what
it's like
pisses Hunter off.
literally experiments that Coonan and other biologists like him are performing that literally refutes this point.
Yeah, they're actually doing the work. They're not
Right.
just saying, "Oh, it can't happen because my mind can't wrap around it."
Because
Which
10
is,
to the 30th power or 20 to the 100th power.
yeah, 20 to the 100th power. It has to go through all those chances before, otherwise it's just half a protein or a tenth of protein. It's not a real protein. The one true protein.
One true protein to rule them all.
Yep, exactly.
How many tries would I need to find a guy in Spain? And I don't know he's in Spain.
Oh,
I just know
Jesus
he's in the world
Christ.
somewhere. Well, I have so many tries. I mean, look, I got decades. I got a lot of money. I can fly around the world. I got a lot of time. I don't have a lot of money, but I'm just pretending here. I got frequent
Oh,
flyer
sure.
miles. I could
Oh my God, he's the Monty Python thing again. We already talked about how it's not like that.
can fly around the world. I can do all these things somewhere. You could have hundreds, you could have thousands of trips. A chemical problem we're talking about.
They do love their
This isn't
analogies, don't they?
a
Yeah, they do. Yeah, dumb analogies which indicate that they haven't really thought any of this through.
problem where, yeah, a few more tries is going to help. By the way, there was a paper in the Royal Society a few years ago that made this argument. They said, look, you know, people talk about this problem of protein evolution. We have more tries than you think. And they actually went through a calculation and they showed, you know, we've got 10 to the 40th tries. We got all kinds of tries here. So you're underestimating evolution. It was a profoundly problematic paper.
I think this paper is called "How Much of the Protein Sequence Space Has Been Explored by Life on Earth" by Dryden. It may be another one, I'm not sure. I went out and found it based on just what he said, but I can't remember which one it is.
First of all, what they didn't realize is 10 to the 40th wasn't enough. So it's kind of hilarious. They thought that was a big number. It's not a big number when you look at the problem of protein evolution.
Again, you're
need
assuming we have to sample the whole protein space in
Yeah,
order to find
yeah.
a functional result.
Yep, we have to look over the entire space. It's just this probability thinking that they're stuck on is just so bad. It's just, and you can't get them out of it. You can't get rid of it.
Yeah.
to the 70th easily, and that's for a simple problem.
Oh, you hear what he said? 10 to the 70th? That's Axe.
the Axe
Douglas Axe. You can tell that these guys all rub shoulders. Well, they're all working together. It's the only
Yeah.
place they can work.
Well, right, because it's only a certain number of them that have produced a certain number of articles on this, so they kind of
Yep.
have to find each other.
Yeah. Yeah, he's associated with let's see, what is
Discovery
it?
Institute.
I think it might be, let me see, is it False Science? Science, and let's see. He wrote an article called False Science. It is for Science and Culture Today. That's his blog. Discovery Institute. Yeah, you're right. Discovery Institute. So
Come to think of it, is Axe even still at Discovery Institute?
who?
Douglas Axe?
Oh, I think he is. Yeah.
Oh.
He's with one of them right now. He's running around shooting his mouth off and getting paid to talk about 10 to the 70th in his really bad paper.
He's still talking about it.
You know, he wrote another paper where they tested a different enzyme with different mutations, and they admit in the paper that 25% of all of the mutations that they did were functional. 25, that's 1 out of 4. That's not 10 to the 70, 1 out of 10 to the 70. That was one of his first papers. Then he wrote this second paper, and then he disappeared from academia right after that. I don't know. Anyway, he got tossed out for something. I forget what it was. He claims it was because God.
Yeah, I was like, I highly doubt it was because of his
expertise.
Yeah.
Or his belief in God or whatever. Yeah.
Yeah.
Yeah. He pissed somebody off over something or fail. He only wrote like 2 or 3. He didn't write that many papers. So he went off and became a talking head. So I kind of want to get paid to lie to people. I don't know. That'd be a good job.
You wouldn't have to do all that studying.
What's that?
You wouldn't have to do all that studying.
Oh yeah. Just skim across the top. All I have to do is learn how to cherry pick quotes.
Read the script.
Oh, I could have AI.
Abstract, you know.
I wonder if AI could. I bet that AI could cherry pick quotes for us that make evolution sound stupid.
It probably could.
It probably could. And they're probably going to start using it. Yeah.
Probably.
For more
They're
complicated
already
proteins,
using it.
it's
Yeah. Okay.
much bigger number. So they were kind of naive about it. But secondly, when they
Real
came up with their
naive.
number of 10 to the 40th, they were assuming the pre-existence of proteins. They needed proteins to make these experiments.
And there we go.
So
Origins are
they're
bust.
trying to solve
Chicken and the egg.
the problem of protein evolution, but their assumption, their model entails the pre-existence of proteins. So it's circular. So yeah, you can't. It's just unfortunately, it doesn't work that way. You can't just say, "Gee, we have a lot of tries."
Because even if you get one, it doesn't matter. You need many.
And that too.
And if you don't have many working together,
And you have to get one at a time, right? So you have to take the dice and you have to roll them 10 to the 70 times. And then you got one protein, but now, oh no, now you need another one. So you got to roll the dice again.
they're all at once, then even if you got one, then the cell is going to die. And so you're going to.
Exactly. It's like having, one cylinder in your car or something. I mean, it's just, there are.
Yeah, there was that rock and there was that one protein that tried to form and then it died. A long time ago, that one protein. Like, Jesus Christ.
Thousands and thousands of proteins, not to mention all the other molecular machines in the cell that have to work together. This isn't just a claptrap of just things floating around helping you to survive. It's a very complicated system that entails many, many, many different entities, molecular entities working together.
He's going to pick on Koonin now.
So what did this Eugene Koonin paper do to solve the problem of protein evolution? They set up a simulation. They called it.
Yeah, it solved the problem of protein evolution. That's what pissed him off, I think, that he thinks that that's what they were claiming that they did. They solved the problem.
Yep.
Protein fold evolution simulator. Protein fold evolution simulator. PFE.
Sorry, or just as Icarus was claiming, it's all irreducible complexity all over again.
Yep. The chicken protein or the egg protein, Icarus. Yeah. Yeah, I remember the good old days when there were lots of proteins. I do. Lots of proteins and some good acid.
FES, in which they said that they simulated the process of evolution. And in that simulation, they're going to see if they can evolve proteins. In the computers, it's all numerical in the computer, but they're going to try and, run these algorithms and show how when you roll the dice and introduce random mutations into a random gene, you can get mutations that will end up with a protein. Not all the time, it fails a lot, but it worked enough that it was convincing. They got enough successes that it was like, yeah, okay, given the time that we have, we could build proteins and we can build meaningful proteins, not just random proteins.
What's he got?
Is he still talking about the Koonin paper, or did he go off onto another?
No, he's talking about Koonin. He's going to pick on Koonin now for the next little while. So anyway, oh my God, I've got like 30 bookmarks left.
I was good. Yeah, I was like, I don't know if we're gonna get through all your bookmark speed. Sorry.
Hey, Demeter, we were talking about you just a little while ago. Flawed model evolution makes it easy to dismiss it as impossible. Next time, could you give me a scientific paper suggesting that a sudden protein arises? Yeah.
Right.
All
anyway. Strong.
Yep. Yeah, I got quite a few here. There's some doozies at the end. Let's see, where am I at?
Yeah, so if you want to pick out your most memorable ones, because I think we got the big point across so far.
Yeah. Yeah, he goes on, let's see, hates proteins, okay, LR contacts, okay, linear contacts, random as, okay, lucky, Rebecca has so many tries, Royal Society, Koonin, say that cheated. Okay, he's I think maybe I got it right here. We'll just play short parts.
predicting what, the structure is going to look like. If that prediction includes more, if it reveals more long-range contacts, save it. And so, yeah, you run that thing for an hour, and the thing slowly gets more and more and more condensed, and you end up with something that looks like a realistic native protein, globular protein.
Yes.
So they knew the destination they wanted to get to, and so they're helping it get to the destination. But natural selection doesn't know the destination it needs to get to.
Exactly. They cheated.
Yep, there he goes. They cheated. Okay, here's how they cheated. They have a criteria. Okay, if you just randomly create and randomly mutate, nothing really interesting is going to happen, right? You're going to be all over the place. You're going to be up, you're going to be down, or whatever. So you have to introduce some sort of selection mechanism or some sort of selection model. And the selection model,
a strong selection pressure that they're modeling.
Right. And the selection model that they used was basically globular proteins or folding, right? That it folded into some structure. And the structures that they got in this paper are pretty fucking cool. I think, where was it? I can't remember. Anyway, I showed you all those different structures that they came up with. They came up with beta sheets and alpha helixes, and they came up with hairpin turns and a whole bunch of other things that we find in proteins. But he calls that cheating, right? And his complaint is that they didn't simulate a real Earth environment and, simulate real organisms with real cellular functionality. How in the hell could they do that? I mean, can you imagine what kind of computer you would have to do that? So he complains about that.
I'm assuming they've based it on like other experiments like that they actually have done with cells. So, and we know how things work, so we can simulate them.
Oh, and the other thing he said, AlphaFold. He says AlphaFold is the AI that's solved millions of structures for proteins right now. He's claiming that because they trained it on real proteins, that it's biased, that it's going to fold according to some bias, right? Yes. Yes. The bias is that's how fucking proteins fold. That is the bias. It's like, it just blew my mind when he said that. I forget what part it is. It's really probably worth listening to if I can find it here. Let's see.
These things are folding into what look like actual proteins, according to them. Maybe they're actually folding into proteins.
Yeah, here he goes. Maybe he said they're not. I don't know. Whatever it is, it's this part right here. It runs for 50. Okay, about 2 minutes. We got to listen to it, I think.
It's a contingency of the paper that is, undercuts everything. Another problem is I mentioned the question of, okay, so you have a mutation, you're going to translate this new gene into a string of amino acids. One of the amino acids changed because you had a mutation. What's going to happen? Is it going to fold up? How much is it going to fold up? What's going to how much? What's the structure going to look like after you introduce this mutation? Well, that alone is an enormous theoretical problem. We don't really know what it's going to look like. We do have some decent tools that predict what that structure is going to look like, but they really only work for native proteins or close to native proteins. So, we've got tens of thousands.
What the fuck is he talking about, native proteins? He's claiming, okay, he's talking about AlphaFold, right?
Right.
And he's going to criticize AlphaFold.
Of examples, proteins that we have looked at, analyzed, and we know the structure, we know the amino acid sequence. And so we fed all that information into AI tools. And so we have these now AI tools that you give it a sequence of amino acids and it will predict a structure. But these tools have been trained on real proteins or
real proteins.
Oh my God. I asked Copilot about native proteins. A native
Yeah.
protein refers to protein in its natural, functional, three-dimensional conformation, the shape it adopts under physiological conditions that allows it to perform its biological role. Key features of a
Okay.
native protein: properly folded, biologically active. It can bind to substrates, catalyze reactions, or interact with other molecules as in, it says as intended. Bad language,
Yeah.
about it.
Yeah,
Stable
bad
under
language.
physiological conditions. Other
Yeah.
forms of proteins are denatured, unfolded, and recombinant.
Yeah. So, okay, he doesn't understand how AI works. He trained it on real proteins. So basically, it looked at real chemical properties and real folds, right?
It's
And
like,
from
yeah,
that,
what else are you going to use to train your AI?
yeah, it's like, facial recognition doesn't work because it was trained on real faces, is what he's saying, right? Or number recognition doesn't work because it was trained on real numbers. Like, what the hell did he expect us to train it on? Like, my
Imaginary
God,
proteins.
we should have trained it on the Bible maybe instead of real proteins. I don't know.
Yeah.
Anyway, oh my
Close
God,
to real proteins. Like they introduce some mutations and they see what happens with the structure. All that information is fed into the AI tools. The AI tools are not trained on the vast universe of all these
they aren't.
sequences that we don't even know about. There's this astronomical number of sequences. What would happen if you had this random sequence? What's it going to look like? We have no idea.
Bullshit. That's what it does. It takes any random sequence and based on the AI training, it predicts the fold. He's claiming that artificial intelligence doesn't work. That's what he's doing. And he doesn't even realize he's doing this. It's just absolutely mind-boggling that he's going on about this. And he thinks that, that was the bias. So because they used AlphaFold, basically, the proteins were biased. I mean, like, what the fuck? Anyway, I don't know.
Yeah.
That was one of my favorites. We could probably lay off here for the night.
I say, do you have any others you want to play real quick?
Oh,
or you think
I
that's
have
the big one?
I have so many. I mean, I couldn't pick. There's just so many.
So, Andrew, can
yeah.
you give me a summary of your observations about Cornelius Hunter and where he strays from the path of scientific credibility?
Let's see. Well, he is knowledgeable about like these different biochemical topics. Like how to put into words. Yeah, he just takes, or well, he does what a lot of them do. He's taking these analogies, he's taking historical context there, he's taking things out of context and using very overly simplistic straw men here. So I mean, it's kind of the standard creationist toolbox in that sense. So
Yeah, so he's just kind of putting together his own mismatch of creationist tropes in his own little
yeah, pretty
style.
much.
Yeah.
I would say so.
And then he talks about how our belief is just theology. It's just a religion. And that's what we believe in. It's like belief in naturalism is theology, and it isn't. Belief in naturalism is what naturalism is how we all live every day of our lives. It's how we make sandwiches.
Keeps using the word theology, and I don't think I feel like he probably knows but he's using it in the wrong context. So.
Yeah, I think he's using the ideas of theology when or religion. He turns theological viewpoints into quote unquote religions when it's also like philosophical viewpoints that aren't religious. Maybe they're worldviews, but like he's talking about naturalism. Well, he's not being nuanced enough to say, well, in science, the way scientists work, it's methodological naturalism, using a mechanistic approach to the world, because that's how you can model things, that's how you can make predictions, and that's how you can make actual tests. It doesn't, you really can't test. God wanted us to chew, so He made us teeth.
Yeah.
And that worldview of democracy, you see, that's just theology.
Yeah.
When you go to it.
Yep. She doubles down on this God can do anything He wants stuff here too, that Sel came up with. Sel said it about 4 times during one of the streams. God can do anything He wants. It's like, okay, that's why it's not science. That's why you aren't thinking. So, there's one part in here. God, they said it just so well. It was
like you
let's see.
And you're not answering the problem in front of you. So what's the point of your answer?
Let's see. Of course, we would have to do. Let's see. We should have Oh, here we go. Okay, this is kind of interesting. Let's see here. I gotta put it back up on the
night.
What's that?
Final clip of the night.
Yeah. Okay.
We don't know how evolution could have done this, therefore evolution didn't happen. Well, we have to decide, there has to be, for us to believe in evolution, there should be reasons to believe in it, right? So that's the problem.
Yeah.
That was it. Yeah, there should be reasons for us to believe in it. How about God? What Rebecca doesn't understand about this atheist is I have never been in my entire life, and I think you're the same, Andrew. Scott, unfortunately, you were raised in the South.
Well, now, I was not raised in really a Christian home. I was raised in a home where Mama was Church of Christ, Daddy was Southern Baptist, and they solved that contention by not taking us to church or teaching us about the Bible or anything. But then I did pick up on that Southern milieu of evangelicalism.
Yeah. Anyway, I have never been given a reason to believe that God did it. So what am I stuck with? I mean, when I look around at all the diversity of life, what am I stuck with? The only thing I've been given a reason to believe in is naturalism, because that's how I put my pants on every day, with naturalism, right?
Honest,
I haven't been.
Seems like you only come up with reasons to believe in God when you already think that God exists.
Yes. Yeah. Anyway, that was wonderful. Rebecca just hit the nail on the head. Yeah, we have to have a reason to believe in God, Rebecca, and you haven't given us one. You keep talking about evolution.
We do have reasons to believe in evolution. You're just not listening to the reasons.
Not listening to the reasons, yeah. Anyway. Okay. Yep.
All right, now, Demeter just asked us to ask AI a question. It's there on your screen, and I just typed it into Copilot and see what they said. Okay, it's too long a response, so we'll have to cover that at a later time, Demeter. I am sorry. It doesn't give a flat yes or no answer. It does mention some paper from Nature in this year. Let me see if I can find that paper.
I think the paper that Hunter was citing was Trust Me Bro 2011.
Okay.
Trust me, bro. Yeah. So I think I did a wonderful thing on the centromere and the fusion problem this morning, and I hope that anybody listening will go listen to that, because I'm going to be really sad if nobody listens to my wonderful discovery about Anchor D, 30BL, and the chromosome fusion, because it's really good evidence for evolution, and everybody should look at that, I think, including you, Demeter. Anyway, Demeter was at a conference, I think. We haven't talked. You were at a conference, right? Ooh, thunder boomies outside. Cool. Okay. I'm ready.
Oh, You think we satisfactorily addressed the video to your liking, Speed?
Yeah, there was so much more to talk about in protein, but I mean, what time is it? Like 10:30. I should be watching Netflix right now.
Yeah, sorry we didn't get to all of it, but
Yeah, it was
No,
a wonderful new.
I did put a link to a Nature article, but I don't think it's actually answering Demeter's question. But it's an interesting title of. Oh wow. It's not that interesting a title.
Isn't it?
Theoester mediated RNA amino acylation and peptidal RNA synthesis in water. So,
cool.
Yeah,
cool.
So it's there in this side chat. Something for you and Demeter to read before you lay your head down to sleep tonight.
Oh, that's by Singh.
Reading it cause your head to go to sleep.
I think I have that one. By Singh. Okay. Anyway.
Thanks, Scott.
Yeah, I think I'm done. Oh, by the way, there's a series, I think it's on Netflix, called Black Rabbit. Maybe it's on HBO. It's who the hell's in it? Everybody should watch it. It's really good. You should watch it.
What is it about?
Scott? It's about a restaurant. A couple of guys that own a restaurant called the Black Rabbit, and it kind of gets pretty fucked up from there. It's with who the hell's
Jason Bateman and Jude Law, so that's a good team up right there.
Yep, it is. Anyway, yeah, it's pretty good.
It's got 8 episodes, and it's on Netflix.
Limited series. Yeah, I started watching it last night. It's pretty hard to put down. Very interesting. Do you watch TV, Andrew?
No, not really.
Restaurant drama has never really been attractive to me. But then again, if drama is drama, it could be about something interesting. Maybe it's the drama that I've seen that's not that great. Maybe this will have better drama, better acting, better storyline.
The description is restaurant drama with a criminal underbelly, and it's the criminal underbelly that's exciting and pretty interesting. So yeah, it is a grabber. It's a good series. You'll like it, guaranteed. Anyway, that's my pitch for the day. And Cornelius, I would love to talk to this guy, but I don't know if I don't know how it would turn out. I don't know. But I'd like to put his nose down in.
I'd like to see that. I don't know if it will happen.
No. Before he would talk to you, he's already said it. You'd have to admit that evolution's a failure.
Oh, that's right. Yeah, he said
people
that.
who admit that evolution's a failure.
Right. Yeah, we have to agree to that first. So, yep.
So in other words, I don't want to talk to anybody because nobody's going to agree to talk to him under those circumstances.
Yeah. When people talk about believing in evolution, I always get this funny feeling that it doesn't apply to me that way. I mean, it isn't like a belief that I have. It's a project that I'm doing. It's really kind of odd, but it always kind of sets me back a little when they say, "Oh, well, you just believe it." It's like, no, I don't just believe it. I keep looking for the next, mechanism, the next secret, the next genome.
Like a book that's still being written.
Yes. Yeah. It's just something that, I don't know.
Yeah.
This idea that, go ahead.
I guess you could say you believe in evolution, but that's not really the process. The process is, well, you accept the scientific consensus because these are the scientists and they do the research and write the papers and review the papers in this topic. If you find some disconfirming information, then maybe you won't believe it. But I'm not saying belief has to be the default position, but why would you just out of hand reject the consensus of the scientists in the field? That's the weird thing. Okay, so let's go to the alternative. Magic man in the sky who can do anything he wants, and there's no reason to think that we weren't all created last Thursday with the world looking as if it had evolved or as if it was advanced age or something like
Yeah,
that.
you see, Scott, it all goes back to their theology in the day of the Epicureans.
Yeah, I go all the way back to that. The thing is, I don't so much believe it is that everything I have, every question I've had, when I start to research it, it looks plausible, right? So everything comes out plausible, like this chromosome 2 fusion thing. They said, "Oh God, there's an anchor D BL30 gene that crosses the fossil centromere," and I'm like, "Holy shit! If that's the case, then evolution certainly does have a problem." So then I go and look at it, and it's like, "Holy shit! I see what happened, and I know that it's plausible now." So they were totally wrong about it being a problem for evolution. It actually is good evidence for evolution.
Jeez.
And it happens that way for everything. Like, how did the liver evolve? I Google it. It's like, oh, fuck, there's all kinds of plausible stuff.
You're
What-
saying all these biologists who trained their lives studying this topic actually have good reason to think what they think? Say it isn't
Yeah.
so.
Yeah, they find that it's plausible because of what they know. And everything that I learn, nothing comes out and says, oh, it could not have possibly happened. Nothing at all. And until I find such a thing, I'm going to just keep thinking it's plausible. Do I believe it? I don't know if I'd put it that way. I think it's plausible.
Yeah.
Anyway, it's an odd thing. I mean, if I just believed it, I would probably stop studying. I don't want to do that. Yeah. Anyway.
Yeah, I think that's a good way of putting it.
Yep. There you go. Okay. I think I want to go and watch the rest of that series. So,
All right.
okay, guys.
All right,
So,
everybody. It was great being on here with you.
Okay. Thank you, Andrew, for showing up. That's
Yep.
Yeah, Andrew, good talking to you.
Yeah, thanks for doing that paper. That was pretty cool.
yeah. Maybe someday we'll sit down with a whiteboard or my drawing pad, and we'll get into this this whole thing with the RNA peptide world and the moment of cellularity, basically when cellularity took off, that sort of thing. Maybe talk about some of that stuff. But and also if you want to compare notes on the genome browsers, I'm sure that we would learn from each other because it's there's a lot in there. Anyway.
Yeah, I just have to find time at some point. Sorry. Yeah, probably sometime in the winter.
Yep. Okay. Okay, guys. I'm gonna end the stream. Thank
right.
you, everybody out there.
All right, good night, everyone.
Goodbye, cruel world.
Bye.